Welcome to the topic of the animal cardiovascular and respiratory systems. The core concepts that we'll cover in this lecture include the dependence of respiration and circulation on diffusion over short distances and bulk flow over long distances, the respiratory function of providing oxygen and eliminating carbon dioxide in support of cellular metabolism, the role of red blood cells in producing hemoglobin, which greatly increases the amount of oxygen transported by the blood, the circulatory system arrangement of vessels of different sizes that facilitate bulk flow and diffusion, and the evolution of animal hearts by natural selection for a higher metabolic rate achieved by increasing the delivery of oxygen to metabolically active cells. Because similar physical laws govern gas and fluid movement, animal respiratory and circulatory systems share many features. As animals evolved to be larger and more complex, they faced the challenge of obtaining an adequate supply of oxygen and nutrients, and at the same time eliminating carbon dioxide and other waste products. Gases, nutrients, and wastes are continually being exchanged between an organism's body and its environment. Because of their small size or thin bodies, single-celled organisms and simple multicellular animals exchange compounds with the environment by diffusion. Larger and more complex animals that cannot rely on diffusion as the sole means of gas, nutrient, and waste exchange evolved respiratory structures at the body's surface for taking up oxygen and releasing carbon dioxide, and internal circulatory systems for long-distance transport to different body regions. The transport of oxygen and carbon dioxide between an animal and its environment is referred to as gas exchange. Single-celled organisms and simple animals and plants exchange gas by diffusion, which is the random movement of individual molecules. Given a large enough surface area, diffusion is an extremely effective way to exchange gases and other substances over short distances between two compartments. Because effective transport by diffusion is limited to short distances, some of the earliest and simplest invertebrate animals were composed of thin sheets of cells with a large surface area. For example, sponges and sea anemones have a thin body with a central cavity through which water circulates, and flatworms have a flattened body surface. When talking about the diffusion of a gas, we usually consider its partial pressure rather than its concentration. The partial pressure of a gas, noted with an italicized P in front of the chemical formula for that gas, is defined as the gas's fractional concentration relative to the other gases present, and this is multiplied by the overall, or the atmospheric, pressure exerted on the gases. Pressure is measured in units of millimeters of mercury. So let's take a look at the partial pressure of oxygen. At sea level, atmospheric pressure is approximately 760 millimeters of mercury. Oxygen makes up approximately 21% of the gases in air. Thus, oxygen in the atmosphere exerts a partial pressure of about 160 millimeters of mercury. For O2 to diffuse from the air into cells, the partial pressure of O2 inside the cells must be lower than the partial pressure of oxygen in the atmosphere. Larger, more complex animals transport oxygen and carbon dioxide over long distances to cells within their body. In addition to diffusion, these animals rely on bulk flow, which is the physical movement of fluid over a given distance. Specialized pumps, like the heart, generate the pressure that is required to move the fluid. Bulk flow occurs in two steps to meet the gas exchange needs of cells in larger animals. The first step, ventilation, is the movement of the animal's respiratory medium, whether that be water or air, past a specialized respiratory surface. The second step is circulation, and this is the movement of a specialized body fluid that carries oxygen and carbon dioxide. In invertebrates, the circulatory fluid is called hemolymph, and it's called blood in vertebrates. This fluid delivers oxygen to cells within different regions of the body and carries carbon dioxide back to the respiratory exchange surface. Gas exchange in complex multicellular organisms can be viewed as a series of four linked steps as shown here. To deliver oxygen to the mitochondria within their cells, animals move fresh air or water past the respiratory exchange surface in the process of ventilation, which is the first part of bulk flow. Ventilation maximizes the concentration of oxygen in the air or water on the outside of the respiratory surface. The buildup of oxygen favors the diffusion of oxygen into the animal across its respiratory surface. Following diffusion into the blood, oxygen is transported by the circulation to the tissues. Internal circulation again serves to maximize the concentration of oxygen outside cells. Oxygen diffuses from the blood across the cell membrane and into the mitochondria, where it burns fuel for ATP production. The same 4 steps occur in reverse to remove carbon dioxide from the body. All animals must obtain oxygen from the surrounding environment. Aquatic animals that take in O2 from water breathe through their gills. Gills are highly folded, delicate structures that facilitate gas exchange with the surrounding water. Some animals, like the tube worm and salamander shown here, have external gill structures, whereas other aquatic animals like this fish rely on internal gills. Terrestrial animals have developed different respiratory structures for removing oxygen from the surrounding air. Terrestrial insects evolved a system of air tubes called trachea that branch from openings along their abdominal surface into smaller airways. Many terrestrial animals such as reptiles, birds, and mammals have internal lungs for gas exchange. The respiratory surfaces of more complex and active animals are highly folded, creating a large surface area within a relatively small space. These surfaces are only 1 or 2 cell layers thick, providing a diffusion distance of as little as 1 to 2 micrometers. Fish need sufficient oxygen to meet the energy demands of swimming. Therefore, fish actively pump water through their mouths and over the gills, which are located in a chamber behind the mouth cavity. Fish greatly reduce the energy cost of moving dense, viscous water by maintaining a continuous unidirectional flow of water past their gills. The importance of this unidirectional flow can be illustrated with the following anecdote. Several years ago, aquarists at the Seattle Aquarium found dead salmon in their large domed exhibit, and the fish showed no outward signs of injury. This strange die-off continued for a few days until aquarists decided to check the nighttime cameras. Lo and behold, an otter that had recently been introduced to the enclosure had apparently been combating boredom by routinely pulling salmon backward through the water. Unfortunately, this disrupted the ability of the salmon to keep water flowing unidirectionally over the gills and thereby suffocated the fish. After that, the otter was removed from the enclosure and placed in his own space. So how do fish maintain adequate flow? In bony fishes, a protective flap overlying the gills, called the operculum, expands laterally to draw water over the gills while the mouth is refilling before its next pumping cycle. Gills consist of a series of gill arches located on either side of the animal behind the mouth cavity and, in bony fishes, beneath the operculum. Each gill arch consists of 2 stacked rows of flat, leaf-shaped structures called gill filaments. These gill filaments contain numerous lamellae, or thin, sheet-like structures. A series of blood vessels brings oxygen-poor blood from the heart to the lamellar surfaces. The lamellae are oriented so that the blood flowing through them in a capillary network moves in a direction opposite to the flow of water past the gills. This type of organization, in which fluids with different properties move in opposite directions, is called countercurrent flow. It is an efficient way to exchange properties between 2 fluids. The property can be a chemical property, such as oxygen concentration, or a physical property, such as heat. Let's consider 2 tubes that are placed next to each other. One is carrying hot water, and the other is carrying cold water. When the water in the tubes flows in the same direction, or concurrent flow, the hot water gets colder and the cold water gets hotter because heat is transferred between the 2 tubes. Given enough distance, the water will be at an intermediate warm temperature at the end of the tubes, reaching the same average temperature in both tubes. Now consider what happens when the water in the two tubes flows in opposite directions, called countercurrent flow. The cold water entering on the left encounters increasingly hot temperatures and, given enough distance, becomes nearly as hot as the hot water entering the other tube from the right. The hot water encounters increasingly cold temperatures as it moves to the left and becomes nearly as cold as the cold water entering the other tube on the left-hand side. This mechanism, known as countercurrent exchange, is used widely in nature. As a result of countercurrent exchange, fish gills can extract nearly all the oxygen in the water that passes over them. At the same time, carbon dioxide readily diffuses out of the blood vessels and into the water that leaves the gill chamber. A set of blood vessels carries the O2-rich blood away from the gills to supply the rest of the fish body. Because of their small size, insects do not have a respiratory surface, but instead employ a direct pathway of air transport that gets air directly to their tissues. Insects rely on a two-step process of ventilation and diffusion to supply their cells with the oxygen and eliminate carbon dioxide. Air enters the insect through openings called spiracles that align either side of its abdomen. The spiracles can be opened or closed to limit water loss and regulate oxygen delivery, much like the leaf stomata of plants. Diffusion occurs at the cellular level, with O2 supplied by the fine airways diffusing into the cells and CO2 diffusing out, where it is eliminated through the insect's trachea. The mitochondria of the flight muscles and other metabolically active tissues are located within a few micrometers of tracheal airways. Because oxygen and carbon dioxide diffuse rapidly in air, the tracheal system delivers oxygen at high rates. Unlike fish gills, the lungs of most land vertebrates inflate and deflate to move fresh air with oxygen into the lungs and expire stale air with CO2 out of the lungs. The low density and viscosity of air enables these animals to breathe by tidal ventilation without expending too much energy. Air is drawn into the lungs during inhalation and moved out during exhalation. During inhalation, mammals and reptiles use muscles to expand their thoracic cavity to draw air inside their lungs. Inhalation during normal, relaxed breathing is driven by contraction of the diaphragm, a domed sheet of muscle located at the base of the lungs that separates the thoracic and abdominal cavities. The muscular expansion of the lungs causes the air pressure inside the lungs to become lower than the air pressure outside the lungs. The resulting negative pressure draws air into the lungs. In most animals, exhalation is passively driven by elastic recoil of tissues that were previously stretched during inhalation. The contraction of the lungs causes the air pressure inside the lungs to become higher than the air pressure outside the lungs. The resulting positive pressure forces air out of the lung cavity. During exercise in mammals, other muscles assist with inhalation and exhalation. The intercostal muscles, which are attached to adjacent pairs of ribs, assist the diaphragm by elevating the ribs on inhalation and depressing the ribs during exhalation. The action of the intercostal muscles helps to produce larger changes in the volume of the thoracic cavity, increasing the negative pressure that draws air into the lungs during exhalation and assisting the elastic recoil of the lungs and chest wall to pump air out of the lungs during exhalation. At rest, humans inhale and exhale about half a liter of air every breathing cycle. This amount represents the tidal volume of the lungs. With a breathing frequency of 12 breaths per minute, the ventilation rate, which is the breathing frequency times the tidal volume, is 6 liters per minute. When more O2 is needed during exercise, both breathing frequency and tidal volume increase to elevate the ventilation rate. The fresh air inhaled during tidal breathing mixes with O2 depleted stale air that remains in the airways after exhalation. This region of depleted air is called the dead space. As a result, the partial pressure of oxygen in the lungs, approximately 100 millimeters of mercury in humans, is lower than the partial pressure of freshly inhaled air, approximately 160 millimeters of mercury at sea level. Consequently, the fraction of oxygen that can be extracted is lower than the fraction that can be extracted by the countercurrent flow of fish gills, which can reach 90% or more. Typically, mammalian lungs extract less than 25% of the oxygen in the air, and reptile and amphibian lungs extract even less. Nevertheless, the disadvantages of tidal respiration are offset by the ease of ventilating the lungs at high rates and the high O2 content of air itself. Despite the limitations of tidal respiration, mammalian lungs have an enormous surface area and a short diffusion distance for gas exchange, and are well adapted for breathing air. Consequently, the lungs of mammals supply oxygen quickly enough to support high metabolic rates. The lungs of most air-breathing vertebrates are blind-ended sacs located within the thoracic cavity. Respiration starts when air is taken in through the mouth and nasal passages and then passed through the larynx. Air then enters the trachea, the central airway leading to the lungs. The trachea divides into two airways called primary bronchi, one of which supplies each lung. The trachea and bronchi are supported by rings of cartilage that prevent them from collapsing during respiration, thus ensuring their airflow meets with minimal resistance. The primary bronchi divide into smaller secondary bronchi and again into finer bronchioles. This branching continues until the terminal bronchioles have a diameter of less than 1 millimeter. The very fine bronchioles end with clusters of tiny, thin-walled sacs called the alveoli, where diffuse gas exchange takes place. As a result of this increasingly branching pattern, each lung consists of several million alveoli, providing a large surface area. Small blood vessels called pulmonary capillaries supply the alveolar wall. Keeping the surfaces of the alveoli moist is critical because moisture helps move oxygen molecules from the air into solution, and thus helps them diffuse across the alveolar wall. Certain alveolar epithelial cells produce surfactant, a compound that acts to reduce the surface tension of the fluid film. Surfactant allows the lungs to be inflated easily at low volumes when the alveoli are partially collapsed and small. It also ensures that alveoli of different sizes inflate with similar ease, enabling uniform ventilation of the lungs. The mucus-secreting cells also trap and remove foreign particles and microorganisms that an animal may breathe in. Beating cilia on the surface of these cells moves the mucus and foreign debris out of the lungs and into the throat, where it is swallowed and digested. Birds must extract oxygen efficiently to supply the considerable energy needed for flight. This has led to the adaptation of a different lung anatomy. The lungs of birds are unique in that they are rigid and do not inflate and deflate. As a consequence, birds do not use tidal breathing. Instead, they benefit from unidirectional airflow that enhances gas exchange by maintaining large concentration gradients for diffusion. A bird's respiratory system consists of two sets of air sacs, one located posterior to the lungs, which means behind them, and one anterior to the lungs, or in front of them. Their respiratory physiology depends on two sets of inhalation and exhalation to move a volume of air completely through the lungs. The first inhalation draws oxygen-rich air into the posterior sacs. Exhalation then moves this fresh air to the lungs. The next inhalation moves the stale air from the lungs into the anterior sacs, and the second exhalation expels the stale air from those sacs. This process allows birds to achieve a continuous supply of fresh air into the lung during both inhalation and exhalation. Animals adjust their respiratory rate to meet their cells' changing demands for oxygen. Respiration is unique in that it is controlled by both the voluntary and involuntary components of the nervous system. In most circumstances, breathing is controlled unconsciously. Homeostasis often depends on sensors that monitor the level of the chemical being regulated. In the case of breathing, these sensors are chemoreceptors located within the brainstem and sensory structures called the carotid and aortic bodies. The carotid bodies sense oxygen and proton concentrations of the blood going to the brain. The aortic bodies monitor their levels in blood moving to the body. Chemoreceptors in the brain sense carbon dioxide and proton concentrations. If the concentration of CO2 in the blood is too high, chemoreceptors in the brain stimulate motor neurons that activate the respiratory muscles to contract more strongly or more frequently. Stronger or faster breathing rids the blood of excess carbon dioxide and increases the supply of oxygen to the body. Breathing can also be controlled voluntarily. Holding the breath makes it possible for humans and marine mammals to dive underwater. Sound is produced by voluntarily adjusting the magnitude and rate of airflow, such as that over the vocal cords of mammals, the syrinx of songbirds, or the glottal folds of calling amphibians, such as some toads and frogs. Once oxygen is pulled from the environment and diffused into the animal, it must be transported through the body. This occurs through the circulation of blood in vertebrates and hemolymph in invertebrates. Mammalian blood is composed of plasma, white blood cells, platelets, and red blood cells. Hematocrit is the fraction of total blood volume that is red blood cells. A human has a hematocrit of 45%, whereas a fish has a hematocrit of 30%. Blood with a lower hematocrit flows with less resistance, but carries less oxygen. Blood plasma is the fluid portion of blood without the cells. It can hold only as much oxygen or carbon dioxide as can be dissolved in solution. How much oxygen goes into solution at a given partial pressure is a measure of its solubility. Because oxygen is about 30 times less soluble than carbon dioxide, only about 0.2 milliliters of oxygen can be carried per 100 milliliters of blood. In both vertebrates and invertebrates, hemoglobin evolved as a specialized iron-containing or heme molecule for oxygen transport. Hemoglobin gives the cells and the blood their red appearance. After O2 diffuses into the blood, it diffuses into the red blood cells and binds to the heme groups in hemoglobin. Hemoglobin's binding of oxygen removes oxygen from solution, keeping the partial pressure of the red blood cells below that of the blood plasma so that oxygen continues to diffuse into the cell. The removal of oxygen from the plasma, in turn, keeps the partial pressure of oxygen of the plasma below that of the lung alveolus. So, oxygen continues to diffuse from the lungs into the blood. By binding oxygen and removing it from solution, hemoglobin increases the amount of oxygen in the blood 100-fold. Myoglobin is a monomer that contains only a single heme group. It is a specialized oxygen carrier within the cells of vertebrate muscles. The percentage of oxygen bound to hemoglobin is called hemoglobin saturation. If we plot the partial pressure of oxygen in the blood against the hemoglobin saturation, as we see here, we get the hemoglobin's oxygen dissociation curve, which has a sigmoidal shape. This demonstrates that as the partial pressure of oxygen in the blood increases, hemoglobin saturation rises slowly at first, then more steeply, and then more slowly again until it levels out. At 100% saturation, all hemoglobin molecules bind 4 oxygen molecules. The increase in binding affinity with additional binding of oxygen is called cooperative binding, and it gives the oxygen dissociation curve for hemoglobin its sigmoidal shape. Many factors affect oxygen binding by hemoglobin. In mammals, the mother's respiratory and cardiovascular systems must provide the fetus with oxygen and remove carbon dioxide. Maternal and fetal blood exchange gases across the placenta, yet the oxygen concentration gradient at this exchange point does not favor the movement of oxygen from the mother's blood to the fetal blood. Fetal mammals are able to extract oxygen from the mother's circulation because, before birth, they produce a form of hemoglobin that has a higher affinity for oxygen than the mother's hemoglobin. The oxygen dissociation curve for fetal hemoglobin is shifted to the left of the curve for maternal hemoglobin. At birth, the newborn's red blood cells rapidly shift to producing the adult form of hemoglobin. Tissue and blood pH have an important effect on the O2 affinity of hemoglobin as well. When pH falls during exercise, that is, when the concentration of hydrogen ions increases, the affinity of hemoglobin for oxygen decreases, resulting in a rightward shift in the oxygen dissociation curve. This phenomenon is called the Bohr effect after Christian Bohr, the Danish physicist who first described it in 1904. Decreases in pH occur when carbon dioxide is released from metabolizing cells, or when an inadequate supply of oxygen leads to the production of lactic acid. Both carbon dioxide release and lactic acid production occur during exercise. Because hemoglobin's affinity for oxygen is reduced, more oxygen is released to the cells for aerobic ATP synthesis. Because there are no interacting subunits in myoglobin, the O2 dissociation curve for myoglobin has a different shape than that for hemoglobin. Over the physiological range of oxygen partial pressure, myoglobin has a greater affinity for oxygen than hemoglobin does, and therefore binds oxygen more tightly. As a result, hemoglobin releases oxygen to exercising muscles before myoglobin does. Once the exercising muscles consume the available oxygen, the intracellular partial pressure of oxygen drops, and the myoglobin releases its bound oxygen. Red muscle cells, which depend mainly on aerobic respiration to produce ATP, store large amounts of myoglobin. The myoglobin in these cells can release oxygen quickly at the onset of activity, before respiration and circulatory systems have had time to increase the supply of oxygen. This graph plots the dives of a beaked whale fitted with a tracking device. Diving marine mammals have large amounts of myoglobin in their muscles. The myoglobin loads up with oxygen when the animals breathe at the water's surface. During a dive, the oxygen from the myoglobin is used to supply ATP. As a consequence, marine mammals can stay underwater for 30 minutes or longer. The O2 carried by hemoglobin is transported to tissues throughout the body by the circulatory system. Generally, smaller invertebrate animals such as insects and many mollusks have open circulatory systems that contain few blood vessels. Most of the circulating fluid, called hemolymph, is contained within the animal's body cavity and bathes the animal's tissues and organs. Open circulatory systems have limited control of where the fluid moves. Muscles that are active in locomotion can assist circulation of the hemolymph, and some invertebrates have simple hearts with openings that help pump fluid between different regions of the animal's body cavity. Animals with open circulatory systems are generally less active. Animals with closed circulatory systems tend to be larger and more active because a closed circulatory system delivers oxygen at high rates to exercising tissues. Closed circulatory systems are made up of a set of internal vessels and a pump, the heart, to transport the blood to different regions of the body. Closed circulatory systems must produce enough pressure to carry the circulating blood to all the tissues. Yet once the blood reaches smaller vessels that supply the cells within the tissues, the blood pressure and flow rate must not be too high. High pressure would cause the blood to flow so quickly through small vessels that there would not be enough time to exchange gases. Closed circulatory systems can control blood flow to specific regions of the body by varying the resistance to flow. For example, wading birds reduce blood flow to their legs when they're in cold water to lessen heat loss. Vertebrates can also increase blood flow to their muscles to deliver O2 and nutrients during exercise. Animal circulatory systems are organized to have blood flow over long distances in relatively few large diameter vessels with low resistance to flow. Arteries are the large, high-pressure vessels that move blood flow away from the heart into the tissues. One easy way to recall the direction of blood flowing through arteries is to recognize that the words artery and away, as in away from the heart, both start with the letter A. Arteries branch into blood vessels of progressively smaller diameter called arterioles. Arterioles ultimately connect to finely branched networks of very small blood vessels called capillaries. It is at the capillaries that gases are exchanged by diffusion with the surrounding tissues. The number of small diameter vessels at each branching point in the circulatory system greatly exceeds the number of large diameter vessels. The reverse organization is found on the right side of circulation. Numerous capillaries drain into vessels of progressively larger diameter, called venules, and the venules drain into a few larger veins that return blood to the heart. The two largest veins, the vena cava, drain blood from the head and body into the heart. There is little pressure available to push the blood forward in the veins because pressure has been lost to the resistance of the arterioles and capillaries. Consequently, veins are thin-walled and have little smooth muscle or elastic connective tissue. Because of the low pressure, blood tends to accumulate within the veins. As much as 80% of your total blood volume resides in the venous system of your circulation at any one time. So how does blood get back to the heart? Well, veins located in the limbs and in the body below the heart have one-way valves that help prevent blood from pooling. The most important aid to returning blood to the heart is the voluntary muscle contraction that occurs during walking and exercise, which exerts pressure on the veins. Above the level of the heart, the return of blood to the heart is assisted by gravity. The organization of blood vessels has two advantages. One, it maintains the same volume of blood flow at all levels within the circulatory system. The increased resistance to flow in the capillaries is offset by the large increase in the number of capillaries. Second, the vessel organization enables the blood to flow more slowly in smaller vessels, providing time for gases and metabolites to diffuse into and out of the neighboring cells. The diameter of a capillary is so small that red blood cells pass through one at a time, and the capillaries must be flexible enough to do that. When an animal becomes active after a period of rest and feeding, blood flow must be increased to its muscles and reduced to its digestive organs. Animals accomplish this using the strong effect of vessel radius on flow resistance. If regions of the body require less oxygen and thus decreased blood flow, arterioles supplying those regions shrink in diameter by contracting the circular smooth muscle fibers in their wall. This reduction in vessel radius increases the resistance to flow and thereby reduces the rate of blood flow. If regions of the body require more oxygen and thus increased blood flow, arterioles supplying those regions expand in diameter by relaxing their smooth muscles, which offers less resistance so more blood can flow through them. A strong pulse of flow pushes against the arterial walls with each heartbeat, about every second in a human. Artery walls are able to withstand these repeated pressure pulses because they contain multiple elastic layers composed of two proteins, elastin and collagen. If an artery wall deteriorates, the collagen and elastin can become so thin that the artery bulges outward. This outward bulge, called an aneurysm, can lead to a life-threatening rupture. Water, certain ions, and other small molecules move from capillaries into the surrounding interstitial fluid by filtration, as shown here with the green arrows. Filtration is forced by blood pressure. Lost fluid and ions are mostly restored through osmosis. As fluid is filtered out of the capillaries, some ions, proteins, and other large compounds remain inside, increasing in concentration. As a result, shown here with the red arrows, there is a tendency of fluid to flow back into the capillary by osmosis. At the same time, blood pressure decreases because of the flow resistance imposed by the capillaries. When the pressure is no longer high enough to counter osmosis, fluid moves back into the capillaries. In vertebrates, excess extracellular fluid is also returned to the bloodstream by the lymphatic system, a network of vessels distributed throughout the body. The fluid that enters the lymphatic system is called the lymph. Small lymphatic vessels merge with progressively larger thin-walled vessels, draining the lymph into lymphatic ducts that empty into the venous system. Lymphatic vessels have one-way valves similar to those in the veins that assist the return of lymph to the circulatory system. A theme in the evolution of vertebrate circulatory systems is the progressive separation of circulation to the gas exchange organ from circulation to the rest of the body. We can begin to see this divergence here with fish. In fish, deoxygenated blood is first pumped to the gills to gain oxygen. Then, the freshly oxygenated blood flows to the rest of the body and returns to the heart. Fish hearts have two chambers, an atrium and a ventricle. Deoxygenated blood returning from the fish's tissues enters the atrium, which fills and then contracts to move the blood into a thicker-walled ventricle. This single circulation path limits the rate of blood flow to metabolically active body tissues. As animals moved on to land, the transition from breathing water to breathing air had important consequences for the organization of vertebrate circulatory systems. Land vertebrates evolved hearts that separated the circulation of deoxygenated blood pumped to their gas exchange organs from the circulation of oxygenated blood delivered to their body tissues. Gas exchange became more efficient and oxygen delivery increased. Metabolic rates rose and animals became capable of greater activity. Reflecting their aquatic and terrestrial lifestyle, amphibians evolved a variety of ways to breathe. The circulatory system adapted through the evolution of an additional atrium, dividing their heart into two separate atria and a single ventricle. In this system, deoxygenated blood from the body, shown in blue, enters the right atrium and is then pumped into the common ventricle. At the same time, oxygenated blood from the lungs, shown in red, enters the left atrium and is pumped to the same ventricle. The resulting mix of both oxygenated and deoxygenated blood, shown in purple, is pumped out of the single ventricle into separate arteries for the lungs and body tissues. This arrangement partially separates the pulmonary and systemic circulations. So, in amphibians, freshly oxygenated blood can be pumped under higher pressure to the body than is the case in fishes. However, because amphibians have a single ventricle, oxygenated blood returning to the heart from the gills or lungs mixes with deoxygenated blood returning from the animal's body before being pumped from the ventricle. Birds and mammals evolved a separate pulmonary circuit to the lungs and a systemic circuit to the rest of the body. This double circulation was made possible by the evolution of a four-chambered heart with separate atria and separate ventricles. As a result, these animals can pump blood to their lungs under lower pressure. This organization has two advantages. It increases the supply of oxygenated blood to active tissues, and it increases the uptake of oxygen at the gas exchange surface. For birds and mammals, metabolic rates increased and endothermic lifestyles became possible. This illustration demonstrates the path of the mammalian circulatory system. The anatomical features are shown on the upper diagram of the heart, while the process is described on the lower diagram. The passage of blood through the right side of the heart is shown in blue on the bottom. Deoxygenated blood enters the right atrium from the superior and inferior vena cava. The superior vena cava returns blood from the head and upper limbs, whereas the inferior vena cava returns blood from the lower systemic capillaries. When the right atrium contracts, the deoxygenated blood moves through an atrioventricular or AV valve into the right ventricle. When the right ventricle contracts, blood is pumped through the pulmonary valve into the pulmonary trunk, which divides into the left and right pulmonary arteries and then goes to the lungs for oxygenation. The walls of the right ventricle are thinner than those of the left ventricle, and its weaker contractions eject blood at a lower pressure. As a result, the blood of the pulmonary circulation moves at a slower rate, allowing greater time for gases to diffuse into and out of the lungs. As shown in red on the bottom, oxygenated blood returns from the lungs through the pulmonary veins and enters the left atrium. When the left atrium contracts, blood is pumped through the left atrioventricular valve into the left ventricle. Oxygenated blood leaving the left ventricle passes through the aortic valve and then flows to the head and the rest of the body through a large artery called the aorta. The thick muscular walls of the left ventricle eject the blood under high pressure to the rest of the body. Heart contractions are coordinated such that both atria contract at the same time, followed by the paired contraction of the ventricles. The full process of atrial and ventricular contractions makes up the cardiac cycle. The cardiac cycle is divided into two main phases based on the status of the ventricles. Diastole is the relaxation of the ventricles. During this phase, the atria contract and the ventricles fill with blood. Systole is the contraction of the ventricles. During this phase, blood is pumped from the heart into the pulmonary and systemic circulations. For the heart to function effectively as a pump, the cardiac muscle cells that make up the walls of the atria and ventricles must contract in a coordinated fashion. Atrial muscle cells must contract in synchrony during diastole to fill the ventricles, and ventricular muscle cells must contract in synchrony during systole to eject the blood from the heart. Cardiac muscle cells are distinct from skeletal muscle cells in two ways. First, specialized cardiac muscle cells can generate electrochemical action potentials on their own, independent of the nervous system. Second, cardiac muscle cells are in electrical continuity with one another through gap junctions, meaning that they can pass their action potentials to adjacent cells. These two features ensure that all muscle cells in a region surrounding a heart chamber are activated and contract in unison. The specialized cardiac muscle cells capable of generating action potentials independently function as a pacemaker that causes the heart to beat with a basic rhythm. These cells stimulate neighboring cells to contract in synchrony. They are found in two specialized regions of the heart, the sinoatrial or SA node and the atrioventricular or AV node. The heartbeat is initiated at the sinoatrial node, located at the junction of the vena cava and the right atrium. When the pacemaker cells in the SA node fire an action potentials spread rapidly from one cell to the next throughout the right and left atria, causing both atria to contract at the same time. Because there is no electrical contact between the atria and ventricles, the ventricles do not contract. Instead, as shown in step 2, the depolarization in the atria reaches a second set of pacemaker cells located in the AV node. A modified set of cardiac muscle fibers transmits action potentials from the AV node to the base of the ventricles. The depolarization spreads throughout the ventricle walls, causing the ventricles to contract at the same time, as shown here in steps 3A and 3B. This delay in transmission from the AV node and through the conducting fibers ensures that the ventricles do not contract until they are fully filled with blood from the atria. Electrodes placed over the surface of the chest and other body regions can record the electrical currents produced by the depolarization of the heart while it beats. The record of this electrical activity is called an electrocardiogram, or EKG, and can be seen here across the bottom of the screen. Cardiologists use EKGs to diagnose heart problems or to determine what regions of the heart has been damaged following a heart attack.